So now we are left with only one structure, that is nucleus. Nucleus is also known as the brain of the cell because it controls all the activities of the cell. It was observed for the first time by Leeuwenhoek. Leeuwenhoek observed it, and he observed it in RBC of salmon fish. In the RBC of salmon. But the credit for the discovery of nucleus and its description goes to Robert Brown, who observed it in orchid cell and he actually described the structure. So discovery, credit goes to Robert Brown, but it was first observed by Leeuwenhoek. Normally, the number of nucleus per cell is 1, normally. So if you talk about number of nucleus, it is invariably 1. If there is only one nucleus, then these cells are known as uninucleate, which is the most common type. But if the number of nuclei is 2, so if there are 2, then it will be called binucleate. One example of a binucleate cell or organism is paramecium. In paramecium, there are 2 nuclei, one larger called macronucleus and one smaller which is known as the micronucleus. If the number is more than 2, so more than 2, then the cell is known as multinucleate. Whenever we talk of multinucleate cells, we also use 2 more terms, syncytial and cenocytic. Syncytial cells are multinucleate cells. There are 2 important things about syncytial cells. Multinucleate obviously, but how did they become multinucleate? It is believed that many uninucleate cells fuse to form a multinucleate cell. Then it will be called syncytial. Example, skeletal muscle. One more important thing about this is syncytial term is normally used in reference to animal cells. Cenocytic, they are formed by multiple karyokinases. That means nucleus originally is one, it divides and it forms many nuclei. Cytoplasmic division does not take place. So originally only one, but by division it is multinucleate. Example, Opalina, which is a protozoan. In fungi, when the septum which separates the fungal cells dissolves, then those septums, when they dissolve, each fungal hypha looks like a multinucleate cell. Then it is known as cenocytic. So fungal hyphae are cenocytic, Opalina is cenocytic. So lower organisms, that is protists or fungal hyphae or these fungi and all, they are considered as cenocytic. Even plants would be considered, if they are multinucleate, would be cenocytic. Syncytial term is normally used in case of animals, and we have also seen how the development of these multinucleate cells takes place. So this is about the number. Let us talk about the shapes. What is the shape of the nucleus? There are various shapes which we have detected. One is a simple spherical, round nucleus which we normally draw. Now here we are drawing a complete wall, but when we see the structure of nucleus in few minutes, we will see that the wall is not complete, it is porous. But just to see the shape. So it is circular. And where do we find these kind of circular nuclei? They are found in cuboidal cells. In cuboidal cells. It can be little oval. So oval shape is seen in columnar cells, the tall pillar like cells, columnar cells. Horseshoe shape, the nucleus is horseshoe shaped in vorticella. Bilobed nucleus. Bilobed nucleus is found in acidophils or eosinophils. Trilobed nucleus in basophils. Multilobed. 1, 2, 3, 4, 5. Multilobed. Multilobed nucleus is found in neutrophils. And a branched or reticulate nucleus, that means the shape is not fixed, but it has multiple branchings. So it is known as branched or reticulate. It is found in the silk spinning cells. Silk spinning cells of insect larvae. So these are some shapes that we have talked of. Very long spindle shape is found in smooth muscle fibers. So there are various shapes. Number is also variable, shape is also variable. What about the size? Size of the nucleus and size of the cell, they correspond to each other, because it is the regulatory body of the cell. If nucleus is smaller, the size of the cell is also smaller. Haploid cell will have a smaller nucleus as compared to diploid and polyploid. So depending upon the cell type, the size will also vary. Let us see the structure of nucleus. It is a double membrane structure, and the membrane has pores. So it is not a continuous sheet like we draw in case of other membranes, and it is a double membrane structure. And at some places we find that this nuclear membrane is in continuation with endoplasmic reticulum. We may not find this connection everywhere, but there are places where we find this extension of nuclear membrane into endoplasmic reticulum. Two membranes, the outer inner membrane, together they make nuclear membrane or karyotheca. Nuclear membrane or karyotheca. Karyon is the term given to the nucleus, so karyotheca. These openings which we see here, these are the nuclear pores. We will draw the structure of nuclear pore in some time. This is nuclear pore. Between the outer and inner membrane, there is a space. Here there is a space, fluid filled space. This is known as intermembrane space. Inside the nucleus is again a fluid very similar to cytoplasm. This is known as nucleoplasm. Also known as karyoplasm, named after the word karyon. In here are found many thread-like structures. These thread-like structures are known as chromatin fibers. The nucleus which we are drawing is an interphase nucleus where these fibers are thread-like. Once the cell starts to divide, then this would condense into a chromosome-like structure. And there is a dense body which is known as nucleolus. We will see all these structures in detail, that is the structure of the nuclear membrane, the pore, even nucleolus, and the chromatin fibers. What is present in this nucleoplasm? This nucleoplasm is rich in divalent ions like magnesium and manganese. Both these ions are required for formation of DNA and RNA. So both are very, very important. Nucleoplasm also has enzymes like DNA and RNA polymerases. DNA polymerase, RNA polymerase. That means enzymes which are required for synthesis of DNA and RNA. How would these things would be synthesized? You would need raw material. What is the raw material for synthesis of DNA and RNA? The nucleotides. So the nucleoplasm also has nucleotides, which is the raw material for production of RNA and DNA, and ions. So basically it is having everything whatever is required for DNA and RNA replication. Now when we come to these individual structures, let us first talk about the nuclear membrane. Let us take all these structures in detail now. The nuclear membrane first. As we said, the nuclear membrane is a bilamellar layer. That means there are two layers, the outer and inner. And we are taking the nuclear pore also, which is present in the membrane. So this is the outer membrane. Outer membrane is studded with ribosomes. So we find certain ribosomes attached to this outer surface. Outer surface means it is in contact with the cytoplasm. So it is also known as cytoplasmic surface of or cytoplasmic face of the nuclear membrane. Inside would be the nucleoplasm. So here we have nucleoplasm. The inner membrane is supported by some fibrillar proteins. This fibrillar protein is known as lamin, and this layer, it is called nuclear lamina. It is also known as lamellar layer, which is made up of a protein called lamin. It is helping in providing strength to this nuclear membrane. So this basically is a supporting membrane, and this space which we see here is the intermembrane space. This gap which is found in the nuclear membrane is known as nucleopore. A nucleopore is not an opening as such. There is a structure which is fitted inside it. It is known as a nuclear complex. So nuclear pore complex is present here. How many pores are there in a nuclear membrane? Normally it varies from 1000 to 10,000 pores per cell. So this is a variable thing. The nuclear pore complex, or this nuclear pore which is visible to us, is octagonal in shape. It is made up of 8 subunits. And the diameter, it ranges from 300 to 1000 angstroms, but the average diameter is about 800 angstroms. So now let us see what exactly is this nuclear pore complex that we are talking of. This is made up of a circular structure and this circular structure has a bulge here, it becomes narrow and again a bulge here. And there are 8 such things. We have drawn 2, there would be one more here. And so total 8 subunits. If we look at the pore from the top, it would appear to have 8 such circular things. And from each one arises a spoke-like thing. So if you look at the pore from the top, it has a structure like this. So basically there is a dumbbell-like thing, the upper end, the lower end, and in the middle there is a narrower end. So it is exposed on the cytoplasmic side also, and it is exposed on the inner side, that is nucleoplasm side also. If you see from the top, it appears just the 8 ones. If you are looking from the top, only the 8 top ones. If you are looking from the bottom, only those 8 bulging things. And in the center, there is a core. Core means again there is a protein which acts as a water channel. So from here the substances can go in and things can come out. For example, the nutrients whatever are required for the nucleus to function, they would go in. When mRNA is synthesized, then it would come out. The substances especially which cannot cross the membrane. So this nuclear pore complex, it is known as nuclear pore, but it is not a simple opening in the nuclear membrane. It is a complex structure which is made up of 8 subunits. Each subunit is exposed on the cytoplasmic as well as the nucleoplasm part, and each one has a spoke which is going in the center. So the lumen is narrower and there is a channel protein which acts as a water channel protein. So substances can go through it. The average diameter we have already written is about 800 angstroms, but the range is there. It is 300 to 1000. So there can be smaller pores also and there can be bigger pores also. So this is a simple thing about the nuclear membrane. Both are same proteins and phospholipid structures, and they are rich in proteins. Now let us come to the things which are inside this. Inside in the sense, the chromatin fibers.